Although it is generally accepted that growth promoters used in the beef industry are not residual in the animal's muscle, are there potentially other risks posed by the common practice of using growth promoters? More specifically, do growth promoters in airborne particulate matter pose risk to human or ecological health? In 2010, USDA's National Institute for Food and Agriculture sponsored research conducted at Texas Tech, Baylor, and Oklahoma State to investigate these questions. This video is a sequel to the project overview produced in 2013 and is intended to summarize the final stages of research associated with this project. Graduate students, under the supervision of faculty, conducted a controlled experimental steroid metabolism and excretion study to further characterize the attributes of growth promoters with cattle at the Texas Tech University feedlot in New Deal, Texas. These data, along with airborne particulate matter data gathered in the previous phases of the project, were then used in a risk assessment model to determine the potential for exposure and adverse effects on human and ecological health. The objective of the Blackwell and Brown study was to develop a time course of fecal and urinary excretion from implanted cattle fed high-energy diets typical of those used on U.S. feedlots, and to quantify degradation of hormones following excretion. This trial consisted of 2 treatment groups of livestock, complete with blood, urine, and feces collection.
When we look at a little bit at the design of this study, we ran into a unique challenge right at the get-go, really trying to find non-implanted cattle in the Texas Panhandle. It's been estimated that 90% of cattle receive an implant at least once in their lifetime, so we really had a difficult time finding non-implanted cattle. So we were fortunate enough to find 16 actually non-implanted cattle, and then we randomly assigned our treatment of no implant or an implant with Revlor XS implant.
When we initially started this project, we were focusing on a set of androgenic steroids, Trenbolone acetate metabolites. These are Trendione, 17-beta Trenbolone, and 17-alpha Trenbolone. As we expanded our work, we included a series of estrogens, estrone, 17-beta estradiol, and 17-alpha estradiol, as well as a progestin called melangestrol acetate. Our research has shown that Trenbolone acetate metabolites are much less common than the estrogen metabolites, and the melangestrol acetate is on similar concentrations with Trenbolone acetate metabolites. Here at the end of this study, we have found that Trenbolone metabolites are excreted largely in the feces. The majority of these excreted are not conjugated metabolites, therefore they are biologically active compounds when they are excreted. We also found higher levels of estrogen being excreted in implanted cattle versus unimplanted.
When we look at serum metabolites, we saw kind of a biphasic release of the parent compounds from the implant. We saw a direct increase in metabolites by day 1 after implantation, and then a slow decrease over time. And we did see a slight increase at day 70 after implantation. This is likely due to just the nature of the implant. Revlor XS has polymer coating on 6 of the 10 pellets, and it's really just designed to slowly decrease or slowly degrade at day 70 and kind of give a secondary dose of Trenbolone acetate and estradiol. When we look at the implications of metabolites, largely serum metabolites, it's believed that there's a threshold for Trenbolone acetate and estradiol to see return performance in those animals. When we really examine our results, we actually found ranges of Trenbolone acetate, largely our alpha isomer, and also our alpha estradiol concentrations were actually lower than previously reported results. And then our alpha estradiol concentrations from our implanted cattle, or our control cattle, were actually higher than previously reported.
Compared to the serum metabolites, the levels being excreted in urine and feces were delayed at the beginning. We saw a large increase by day 3, whereas in serum we saw an increase at day 1. After this increase at day 3, the excreted levels stayed elevated throughout the life of the implant. There was some correlation between serum levels and excretion levels.
As this project draws to a close in 2014, the research team is developing a risk assessment model focusing on specific issues in geography and climate unique to arid and semi-arid production regions. A risk assessment model consists of 2 components, an assessment of exposure as well as an evaluation of the effects of exposure to human and ecological health.
In terms of an overall risk assessment, this excretion study helps us to better understand the levels being excreted from cattle on the feed yard.
In this excretion study, data for determining exposure was collected, and more definitive values of steroid metabolites excreted per head on a daily basis by feed yard cattle was determined. The excretion study data can now be evaluated against steroid levels in particulate matter from feed yards, determining the amount and rate of degradation between the 2 evaluation points. The excretion study portion of the project, along with other studies in scientific literature, provides exposure data, but there are few studies on the effects to human health. With the data produced by the excretion study, researchers can develop models predicting levels of steroids in particulate matter moving off of cattle feed yards, and risk assessment modeling will account for the uncertainty of the effects to human health.
To better understand the effects in human exposure, some collaborative work with the pharmaceutical companies developing these compounds, as well as human health science physicians, and then those who work in particulate matter exposure especially. With these groups together, we would be able to much better define the potential risks of these compounds in particulate matter to humans. Now, the risk of particulate matter itself without steroids is fairly high in humans. So the addition of steroids in small particles may not add considerable risk to the particulates themselves.
The use of growth implants is common today in many stages of beef production, and projects like these help to protect agricultural workers, communities, and our environment from unintended consequences of technologies and management practices which have made American agriculture a productive and efficient industry. These projects are only possible when federal funding agencies, such as USDA's National Institute for Food and Agriculture, provide the funding and oversight that enables those in production agriculture to work hand in hand with scientists and outreach specialists.